Hey everybody, this is for everybody who missed the yeast lab, or maybe you were there and you aren't totally sure how to answer that CER at the end. We're gonna go through that. All right, so let's start with a few background basics you're gonna need to know in order to get through this lab. The whole purpose of this lab is to support a claim with evidence that we will gather to show that cellular respiration has occurred. Let's back up. Okay, so we're going to use yeast for this. Yeast is a very important single-celled organism. It's actually a fungus, so like a kind of mushroom. And yeast is very important to industry. Lots of things are made using yeast, and most importantly, pizza. Okay, so yeast does cellular respiration. So remembering that cellular respiration is the process that cells do where they get energy from sugar. Here is the chemical equation. C6H12O6, that's sugar. And oxygen makes carbon dioxide and water. So up here, anything to the left of the arrow, so the glucose and the oxygen, these are our reactants, the things you're starting with. And then these two things down here, the carbon dioxide and the water, those are our products. That's what is made from this reaction. So left of the arrow, reactants, right of the arrow, products. So this is the process that yeast does. It's called cellular respiration. Byproduct of this, or the byproduct is probably not the best word, it's the reason it happens, is it makes energy. And it's energy for all those other things that the cell's got to do. Grow, reproduce, all of that is provided from this process. So we are going to be looking at yeast, sugar, in a solution. And we are going to provide evidence in our experiment that cellular respiration has occurred. So we got to know about two other things. One of them is called bromothymol blue. I'm just going to shorten it to brom blue. Now, you've seen this stuff before in the exercise lab. That was the stuff that we mixed in the water. It was blue, but then we exercised and we blew into it. It turned yellow. So brom blue is yellow when there is carbon dioxide. So this is an important thing to know. It turns yellow in the presence of carbon dioxide, our product. Now here's a new one we haven't used before. It's called methylene blue. Now methylene blue is also, blue, but it is only blue in the presence of oxygen. So it's only blue when there's oxygen. When there's not oxygen around, it's just white. It's just clear. It has no color. It's the color of the rest of the solution. All right, now I'm going to get my little lab assistant and our supplies, and we're going to set up the lab so you can see what it looks like before and after. Okay, here we are. Got my trusty lab assistant here.
Aye.
And we're setting up the experiment. So, yeast needs water, and the water has to be warm but not hot. You don't want to scorch your yeast and kill it. It's very sad. I scorched my yeast once trying to make pizza. It was hard as a rock. Very depressing. Okay, so we got some water, and this water has the bromothymol blue in it already. So I'm gonna pour that right in here. This water, here, put a few drops of this. This is the methylene blue. So bromothymol blue, methylene blue. 3-4 drops. Let's see what it looks like. Now notice we're not really measuring. You don't have to.
2, 3.
That's good.
Cool.
Let's get it out of swirl. All right.
I like that color.
All right. So now, Aiden, we need, this is just plain sugar.
Aye.
Give it each a scoop into each one.
I should probably do this.
Okay.
Pause the video if you want.
No, that's good. I think you've beaten it enough. Not a big scoop, small scoop. We don't need a lot.
How much?
That. Okay, you do this one. So, in order for cellular respiration to occur, we've got to add some sugar into the solution. Perfect. Dump it in. So I'm just going to give each one a swirl. All right, and now here's where we're going to take our beginning observations. If you look at your lab report, you're going to see there is a table, and I have put a place in the table for your before and your after observations. So bromothymol blue, methylene blue. Remember, please, that I hate it when you put one word for an observation. Not yet, please. All right. Be a little more descriptive than that. Blue will be marked incorrect. Okay, so here we go. Bromothymol blue, methylene blue. Okay, so now we're gonna put a scoop of yeast into each one. Okay, and then we're gonna cover it and we're gonna wait about an hour, and I will come back and finish the video with you so you can see what it looks like then. All right, a scoop, a scoop.
This much?
Looks good. In it goes. Into the methylene blue,
And this one?
and then into the bromothymol blue.
Give it a shake.
We're gonna give it a swirl. And now the important thing is to not touch it after this, because you don't want air getting into it. So I'm gonna cover these with some plastic wrap, and we're gonna leave them for about an hour, and then I will show you guys what it looks like, and we'll discuss the CER at the end. Okay, so it's been about an hour, and here's our lab now. And just a reminder that this one had the bromothymol blue, and this one had the methylene blue. And otherwise, they were identical. They had the sugar, they had the yeast. Now, the first thing I want you to see is look at all of this foam. So think about the process of cellular respiration. Think about what we've discussed in terms of that's what we do, and we're breathing out carbon dioxide. So what do you think that means for the processes happening in these flasks? And then I want you to look at the color changes. So bromothymol blue, think back to the exercise lab, go back to the beginning of this video, read the top of the lab where the instructions are, and I told you about what the color change of bromothymol blue means. And then I need you to do the same thing for the methylene blue, which you see they've both had pretty drastic color change here. So now the last thing you have to do, once you've filled in your observations, is go to the bottom of the lab, where I ask you to support a claim with evidence about that, to say that cellular respiration has occurred here. And you should be talking about both of these flasks in your responses. Now, to help you along with making sure that your message is clear in your response, you could say something like, the color change observed with the bromothymol blue indicates, and the color change observed with the methylene blue indicates. Again, check the top of that lab, or go back to the beginning of this video where I told you what the color changes would mean. And why were they blue and why are they not blue now? Okay? All right, people, I'll see you in class if you have any questions.